Hi, in today's video we are going to talk about Helicobacter pylori and the application of the Rep-PCR test for its genotyping. As a first glance, we will review basic information about the bacteria, starting by its microscopic characteristics. Helicobacter pylori is a gram-negative bacterium with spiral and bacillary morphology, characterized by having 4 to 6 unipolar flagella. This flagella is protected by an extension of the outer membrane of the bacteria, acting like a sheath. Thanks to this, Helicobacter pylori can inhabit and colonize the human stomach, since its flagella are capable of resisting the attack of stomach acid and peristaltic movement. Going more in depth about the history of this bacteria, we can go back to studies that were carried out in patients with gastrointestinal infection with this bacterium. It was found that the most frequent histopathological diagnosis associated with Helicobacter pylori is chronic non-atrophic gastritis in 36.4% of all cases, whereas two other pathologies, such as cancer and gastric ulcer, are also associated but in a lower percentage of 9.3 and 5.1% respectively. The natural history of chronic non-atrophic gastritis caused by Helicobacter pylori, according to Levin and Clark, describes it since it develops from the initial exposure to the bacterium till the disease progression. The pathogenesis of this disease includes 2 stages. The first one is characterized by the arrival and penetration of the microorganism into the gastric mucus, where it settles and multiplies. At this stage, the bacterium releases several toxic substances that are able to stimulate the local immune response, expressed in an increase in secretory IgA in order to prevent the infection process. The main inflammatory cells involved in this initial process are neutrophils, which are attracted to the site of injury. Hence, their presence in the company of lymphoid follicles is considered a sign of activity. In the second stage, there is an amplification of the inflammatory response caused by chemical mediators released by the cells of the immune system, the enteric nervous system, and the complement system that perpetuate the inflammation. Without intervention, this condition can predispose to complications such as peptic ulcers or gastric cancer. For this reason, the importance of early detection and treatment to prevent the progression of the disease and its complications is highlighted. Now, to explore how it is possible for this microorganism to colonize and generate the associated pathology to it, it is crucial to signal the virulence factors that help it. This bacteria can reach its objective by 1. Having urease activity, which promotes the liberation of ammonia that could induce damage on the gastric epithelia. 2. Having bacterial adhesins that are fundamental for the colonization process. 3. Possessing hemagglutinins that could induce autoantibodies due to their biochemical similarity with some antigens of blood groups. And 4. The presence of vacuolization-associated gene, VAC-A, and cytotoxin-associated gene, CAGA, that correlates with highly virulent strains exhibiting toxic activity.
Now, let's move on the Rep-PCR test. This is an epidemiological evaluation test for different diseases, which is based on the fact that some bacteria have short repeated sequences known as palindromic extragenic repeats. Helicobacter pylori can cause diverse gastrointestinal diseases, mainly gastritis, and a lower percentage of gastric ulcers or gastric cancer. This variation can be attributed to different strains, which is why Rep-PCR test is used to identify if there are fingerprints associated with a particular strain. What are these fingerprints? The fingerprints read for Helicobacter pylori can vary depending on the study. However, the ones most commonly studied are those of BK gene. The BK gene has a structure with around 100 to 2,000 amino acids, with 3 regions: the N-terminal, the middle, and the C-terminal. In the N-terminal region, the single sequences are found and can be of type S1 and S2, and in the middle region the types are M1 and M2. According to the variation of the sequence signals, the presence and degree of vacuolizing activity can be determined. S1 type sequence in the gene has vacuolating activity. Region M1 has more activity. The main activity used is the synthesis of vacuolating cytotoxin, which is secreted by the type 4 secretion system and enters cells through endocytosis. Among the biological activities it performs, the main one is the induction of cellular apoptosis, either due to its accumulation in cellular compartments or due to the dissipation of the mitochondrial transmembrane potential. Likewise, it alters the junctions of epithelial cells and inhibits the proliferation of T cells. And how does the test work? It is necessary deionized water, a buffer, nitrogen bases, magnesium chloride, and a Taq polymerase. Primers will vary depending on the sample to be analyzed, since it can be done from the genes PEG-A, CAT-A, URA, etc. Some examples of primers and their sequence are seen in the following tables. The sample for the test is obtained from clinical samples, which are grouped into isolates from patients with gastritis, gastric ulcers, duodenal ulcers, gastric carcinoma, and lymphoma. The sample is obtained through a gastric biopsy. From this sample, cultures are carried out on dent plates and BHA agar. To identify the bacteria to be studied, a gram stain is performed and positive tests are obtained for urease, catalase, and oxidase. Then we proceed with the extraction of genomic DNA. To lyse the bacterial cells, they are washed with sodium chloride and centrifugated. The cells are suspended in trees and EDTA and incubated with lysozyme and rNase A. ASTase, sarcosyl, and proteinase K are also added. The cell's lysates are extracted with phenol and chloroform and precipitated to genomic DNA with ethanol. Genomic DNA is quantified using DNA spectrophotometry, and the integrity of the genomic material is evaluated using agarose gel electrophoresis. The sample for PCR is prepared with the sample with genomic DNA, the two chosen primers, the nitrogenous bases, and the Taq polymerase. Then the amplification is carried out with a thermocycler where the different phases occur: initiation, denaturation, alignment, and extension cycles, and the final extension. The samples obtained after PCR are mixed with tea and stained with ethidium bromide, and then placed on agarose gel. To evaluate the results, rather than a positive or negative control, controls of strains obtained from patients with conditions to be evaluated are used. Here are some examples of positive controls as well as negative controls. In such a way that a test is considered positive when the bands of each sample agree with the control, and it's negative when they do not comply with it. Some advantages of the test is that the microorganisms can be detected regardless of the viability of the bacteria in the samples. The test evaluates the presence of the bacteria, and antibiotic resistance test can be performed. Disadvantages include limited availability, high cost, and use of a specific material. But what are the costs for Rep-PCR test? The general PCR test and the Rep-PCR test are very similar, so it can be considered that they will present similar costs. But one of the main differences is the primers used in Rep-PCR test, since its extra cost ranges from $9 to $15 per base pair. However, it must be considered that the cost varies depending on the number of samples, type of samples, and oligos to be used.